Okay. So last time class, we talked about DNA, right? DNA. So do not forget this. DNA are our carrier of the hereditary traits. We have 3 types of DNA. And if we are going to talk about the concepts, do not forget when you say antiparallelism, remember that DNA has 2 strands, double helix tayo, di ba? The first strand will start at 5' and end with 3'. This 5 and number 3 are referring to the carbons wherein there are attachments between not carbon, phosphate and the sugar. Okay? Di ba ang backbone ng ating DNA are alternating phosphate sugar. So 5' magsa-start ang isa, mag-e-end sa 3'. But the other strand will be anti-parallel. So it will start at 3'. The link between the first phosphate and the first sugar will be at third carbon, kaya 3' siya, and it will end sa 5'. So yan yung tinatawag nating antiparallelism. And then complementarity, because we have 2 types of nitrogenous bases, the pyrimidine and the purine. And if you are going to look at the sequence, kailangan magka-partner talaga purine and pyrimidine natin. So, merong equivalent pair since partner si purine and si pyrimidine. Kung ilan lahat yung pyrimidine sa isang strand, ganun din karami yung purine. Okay, that's the meaning of equivalent pairs. Now, we talked about central dogma of molecular biology. We have 3 processes here. The first one involving the DNA. We need to copy our genetic material. The DNA can do replication. Now, if we want to produce proteins, then the DNA can be used first to produce the RNA. The process is called transcription. And then from RNA, we will be going to make polypeptides or our proteins. And then the process again is called translation. So the proteins are very essential sa life. Without proteins, there will be no life. So even our own cells are made up of proteins. Okay, so we have here, again, the DNA to DNA replication, DNA to RNA transcription, RNA to protein translation. Pero, there are species that are able to do this so-called reverse transcription. Okay? Reverse transcription is when the RNA is being used to produce DNA. Baliktad. So again, reverse transcription. And then I have mentioned this last meeting, that is the HIV. The human immunodeficiency virus has the ability to do reverse transcription. The HIV is actually an RNA na virus, pero when it enters our body, the RNA will do reverse transcription and mag-produce siya ng DNA. Pag si DNA na-produce na, ma-incorporate yan siya sa sarili nating DNA. That's how infection happens for the HIV. So HIV lang yan haba. If the condition becomes terminal, that's the time we call it AIDS. So if magpaparami tayo ng RNA replication is also possible. Okay? So we talked about this enzymes last time involved in the replication. So a little review, since the DNA is double helix, we need to unwind it first, and that is the responsibility of the helicase para mag-unwind, para maghiwalay yung double strand. Now, to start the replication, we need a primer. And for us to create a primer, we need the enzyme primase. A primer class is just a short piece of DNA na ginagamit as a starter for the replication. Now, since the 2 strands of the DNA have the ability to recoil, so na-uncoil na yun using helicase, pero mag-coil sila ulit to form the double helix. Pero kailangan natin yun siyang i-prevent kasi pag nag-recoil yun, walang DNA replication na mangyayari. So the enzyme responsible to prevent the recoiling is the topoisomerase. In bacteria, we call that DNA gyrase. Okay? Now, the major enzyme responsible for the DNA replication are the DNA polymerases. So marami yan sila, 1, 2, and 3. They can do replication starting from the 5' to the 3'. And they can also do proofreading, meaning babalikan nila yung ginawa nilang DNA to see if merong mali. Okay? Kasi pag may mali sa na-copy nila, automatic, that is called mutation. Okay? Only the DNA polymerase 1 has the ability to remove the RNA primer. So the rest, wala. I hate this. Okay, so nasabi ko na ito kanina. And then last one, we have the DNA ligase that will join the fragments of the lagging strand. So pag sinabing lagging strand, meron tayong leading strand and then lagging strand ha. The leading strand, ayun yung tuloy-tuloy yung pag-copy niya. Kasi ano yun siya, primary strand ng DNA yon. But the other strand class, pag kokopyahin na siya, lagging siya, meaning may mga hindi siya diretso-diretso. Naka-fragments lang siya. Ang tawag sa maliliit na fragments na yon ay Okazaki fragments. So, para mabuo talaga yung whole na DNA strand, magdikit-dikit yung mga fragments, we need the DNA ligase. I hope that is clear. So pag sinabing genes, that's just a fragment of the DNA, a part of the DNA. And this one includes all the things na nakopy na doon, no? Yan yung makikita natin either sa ating physical appearance or kung hindi man natin siya makita, andun pa rin siya, but we can pass it on to the next generation. Okay? So, mutations class only occur in the DNA. So, sa ating genes, magkakaroon yan ng mutation. Now, let's talk about transcription since we are done with the replication last time. Pag sinabing transcription, it's making an RNA from our DNA. And saan ito nagtitake place? Sa nucleus pa rin. So just like the DNA replication, sa nucleus pa rin ito nangyayari. Now take note of our 3 types of RNA, mRNA, tRNA, and the rRNA. So ano itong tinatawag na mRNA? Ito yung ating template for protein synthesis. Okay? Ito yung galing class sa DNA replication. So, I hope that pinanood nyo yung video kanina, nakita natin doon, from DNA through the RNA polymerase, gagawa ng strand ng RNA. So, yun yung tinatawag na mRNA. Yun yung template natin for protein synthesis. mRNA means messenger RNA. Pag nabuo na si messenger RNA, it has the ability to escape the nucleus through the pores. Pores natin sa ating nuclear membrane. So mag-exit yan siya doon, pupunta siya sa cytoplasm. Kasi translation or the protein synthesis happens sa cytoplasm na. Okay? Now, itong mRNA, ito yung nagko-contain ng tinatawag na codons. So pag sinabing codons, ito yung sequence, tatlong nucleotides. Excuse me. Tatlong nucleotides na sequence, tatlong letter siya. So tatlong nitrogenous bases. So yung isang codon class will code for a specific amino acid. Again, tinatawag natin yon na codon. Now, pag si mRNA has now escaped the nucleus, nasa cytoplasm na siya, magkakaroon na ng translation. And ito na ngayon papasok si tRNA. So it is the adapter molecule now for our amino acid. And ito yung nagka-carry ng ating anti-codons. So ito yung pinapagawa sa inyo sa biochem dati. Katulad ko tutulan. Where is my pen? Pen. Lagi ko kasulat. Alat kayo akong world. Okay. Yung pinapagawa sa inyo sa biochem class na wait, ay, ayan pala. Ay, okay. Ayan na. Again, written code yung pinapagawa sa biochem na for example, you have this one. Ay, lang time means ano. Okay, ito yung example, ito yung ating codon. Nandito yun siya sa mRNA. Pag kokopyahin na siya, 'di ba? Pinapagawa ito sa biochem ninyo. Ano bang partner ng A? Since this is RNA, ang partner ng A is U. You cannot see thymine for the RNA. And then this one is A, and this one is C, and so on. So pag kinopya na siya, the mRNA, this is now the so-called tRNA. Ito na ngayon yung pinatawag na anti-codon. So that is still a sequence of 3 nucleotides complementary. So, kapartner siya doon, complement siya ng existing codon. Okay? So, ginagawa yan siya when we do translation, yung gagawa na siya ng protein. So, meron yan siyang bit-bit class na amino acid. So, kapag mag-join sila together, yung amino acid nila magdidikit-dikit, and that's the time we get the polypeptide. And then lastly, the rRNA is the one making up the ribosome. So do not forget the one that makes the protein is the ribosome. You can see that in the video na unang didikit doon sa mRNA yung small complex ni ribosome. Tapos pag andun na after that, yung malaki naman dito sa taas na meron siyang tatlong parts, the EPA. So, dyan, nandito yung ating codon. So, papasok na dyan ngayon yung mga anti-codon. Pero the one that makes up this whole thing, the ribosome, is called the rRNA. Any questions so far? Baka may hindi clear sa sinabi ko. Wala. So again, if the DNA polymerase is the one responsible for the DNA replication, pagdating naman class sa transcription, you have there the RNA polymerase. Pero unlike the DNA polymerase na kailangan niya muna ng primer, kailangan niya muna yung short DNA as a starter for the replication. This time, ang RNA polymerase hindi na yon kailangan. Does not require a primer and initiates polymerization of the DNA. Okay? And then does not possess proofreading activity. Unlike our DNA polymerases na babalikan niya yung trabaho niya, si RNA polymerase hindi. So again, hindi siya nag-proofread. Nung gumawa siya ng RNA strand, ayun na yun, diretso na. Kukunin na lang yung mga introns. And then that's it. That's the messenger RNA na walang proofreading na mangyayari. Now, kapag again, nasabi ko kanina, if the mRNA exits the nucleus, translation happens in the cytoplasm. So you have there the mRNA partnered with the tRNA to give us the polypeptides. And then you saw this in the video. In translation, there are 4 steps. We have the activation, initiation, elongation, and termination. Sa initiation, protein synthesis happens if there is the presence of the start codon. So remember the codon sa mRNA, pag nakita na si AUG, nag-appear na si AUG which codes for methionine, magsa-start na yung protein synthesis. So do not forget that our start codon is AUG. Ano yung kinocode niya na amino acid? That's the methionine. So initiator yan siya ng protein synthesis. Okay? Now, sa elongation, ayun yung repeat lang na process na you have succeeding codons, magpapahaba lang tayo ng polypeptides after the appearance of the AUG. And then we have the termination. So, termination happens if there is now the presence of one of the three stop codons. So, tatlo yung ating stop codons sa uga, uwa, a, uwag. So, whatever comes first. Again, uwag, uwa, a, uga. That's our stop codon. Bakit sila tinawag na stop codon? They don't correspond to any amino acid. So, definitely, masa-stop talaga ang protein synthesis. So unlike all the other codons, just like kanina, AUG codes for methionine, other codons code for other amino acids. Itong tatlo, wala silang dala, wala silang kinocode na amino acids. So mataterminate yung ating protein synthesis. Now, if this one, any of this stop codons appear, there will be now the so-called release factor that will bind with the stop codon. Magbabind siya doon sa stop codon and magdi-dissociate na yung lahat ng complex na yon. When I say complex, yung ribosomal subunits natin, the small ribosomal subunit and the large one. And then, ayun na, mag-e-end na ang ating protein synthesis. Okay, so for eukaryotes, protein products will go to our Golgi or Golgi apparatus. Sa ribosome, ribosome yung mag-produce or magsisynthesize ng proteins. Pero once the polypeptides are being released, pupunta siya sa Golgi apparatus either for storage or for some modification. So merong mga modifications na mangyayari. Ano ibig sabihin ng modifications? Pwedeng si polypeptide will be attached to a carbohydrate. So magiging ano na siya, glycoprotein na or mucoprotein na siya. Ganun yung mangyayari. Or kung hindi siya sa carbohydrate mag-a-attach, pwedeng sa metals. Pwede din naman sa lipids. Depende kung anong process ang i-undergo niya. Yan yung sinasabing modification that will happen sa Golgi apparatus. Okay? So let's compare the transcription and the translation processes between prokaryotes and the eukaryotes. Prokaryotes are our bacteria. We have here transcription and translation happen at the same time. So sabay yung transcription and translation ng mga bacterial cell. But for eukaryotes, just like us, ang mangyayari, transcription muna, kokopyahin muna si messenger RNA with the tRNA, ay ano muna, DNA to RNA muna and then RNA to protein. So, transcription first before translation. Take note that higher errors in transcription than in replication. What's the meaning of this? Kasi, diba, si DNA polymerases can do proofreading. But yung RNA polymerase, hindi niya kaya mag-proofread. So definitely, there will be higher chances of errors in transcription than in DNA replication. Okay? So I hope that is clear. Now, for let's define this term degeneracy. If you hear this term degeneracy, when it comes to biochem, it means that in one amino acid, there will be many codons. Doon sa video, I'll give you a copy of this. I just don't know if I can really post this as Schoology. Baka mabigat yung video, but let me try. But, again, class, nandun sa video yung table ng mga amino acids that will code for a specific, nandun yung list ng codons that will code for a specific amino acid. So just like serine, si serine class, hindi lang isa yung codon na pwedeng mag-code sa kanya. Marami siya. So ang term na yan is called degeneracy. So kahit hindi mo makita yung isang specific codon, meron ka pa namang other codons that will give you the specific amino acid that you need. Again, yan yung ibig sabihin ng degeneracy. Now, let's talk about mutations. If there is a change in one codon in our mRNA, that is when point mutation happens. So, isang codon lang isang codon lang yung na-change automatic, that will be a mutation. So, ang tawag niyan, kapag ang type of point mutation is called transitional, it means a purine is being replaced by a purine. So, anong ibig sabihin? Originally, for example, you have this one sa codon. Now, kapag nagkaroon ng mutation, and then naging ganito siya. This one is the correct one. Anong naiba? Itong G. Instead of A, naging G siya. But take note that both of them are purine bases. Okay? So yung purine, napalitan siya ngayon ng another one, pero purine pa rin. Ang tawag niyan, transitional point mutation. So that is also true if a pyrimidine or a pyrimidine is being replaced by another pyrimidine. Example, ito pa rin. Pero this time, A pala A. Okay. So ito, cytosine, supposedly cytosine siya originally, pero na-replace siya ng uracil. Okay? Again, nag-replace ng uracil dito. This is still pyrimidine. Both of them are pyrimidine, so transitional point mutation yan siya. Pero kapag purine to pyrimidine or the vice versa, we call that transversional. Okay? So we have here, for example, CAG, and then naging CCG siya. So instead of adenine, nagkaroon ng mutation, naging cytosine. So this one is a purine base, but the cytosine is a pyrimidine one. So purine, pyrimidine, yung naging mutation, we call that transversional. I hope that is clear. So isang nitrogenous base lang ang napalitan dyan, automatic mutation na ang tawag. Okay? Questions or clarifications? I hope nakuha yun na. Now, consequences. There are consequences for these mutations. Meron tayong tinatawag na silent mutation if the changed codon codes for the same amino acid. So kagaya, sabi ko 'di ba kanina na for example si serine, maraming nagko-code sa kanya na codon. Ngayon kung nagkaroon ng mutation, pero pag-mutate, yung lumabas na codon still codes for serine. Again, example lang yan ha, the same amino acid pa rin yung kinocode niya. Ang tawag dyan silent mutation. Merong mutation, pero hindi mo siya makikita kasi the same amino acid pa rin yung kinode ng pumalit na codon. So I hope that's clear. That's silent mutation. Now, we have the so-called missense mutation. If the changed codon codes for a different amino acid, ito, iba na talaga. Nag-change yung codon, nagkaroon ng mutation, tapos yung na-change na codon, codes for a different amino acid. Ayun, makikita na talaga natin yung mutation for that. And then we have the nonsense mutation. If the change codon codes for a stop codon, so nag-elongate pa lang yung polypeptide, hindi pa siya tapos supposedly sa protein synthesis, but biglang nagkaroon ng mutation and the codon class is a stop codon. So yung na-change na codon, biglang nag-UGA siya or yung UGA. So automatic yon, masa-stop ang protein synthesis and it will become nonsense. Kasi hindi pa nga tapos yung ating protein synthesis. Hindi pa siya magagamit kasi kulang pa. Hindi pa siya nag-elongate sa target niya talaga para magamit siya as anything, kung ano man ang gamit ng ginagawa na protein na yon. So, nonsense na ang tawag doon. Get nonsense mutation. And then we also have frame shift na mga mutation, either insertion or deletion. So si insertion, para siyang point mutation pa rin. Ano pa rin siya? Either translational or transversional. So kagaya nito, napalitan, instead of C, naging A siya. Deletion kapag ka nakulang. Pwede din yun. So supposedly si codon, 3 dapat yan siya. Pag may nakulang dyan, we call that deletion. So all of them class are mutations. So either makikita mo physically or pwedeng wala tayong makita. Pero meron na palang mutation na nangyayari. Okay? So, na-mention ko itong phenotype and genotype. Phenotype class, ito yung mga physical traits and yung appearance mismo natin na nakikita natin with our naked eye. So, just like me, I'm very much similar to my father. Like ang ginatawag ko nilang like carbon copy or photocopy of my father. This is my phenotype. Ang genes class ng father ko is lumabas sa aking appearance. We call that again phenotype. So lahat, pati physical traits. Now, yung genotype is the sequence of the genome. So it means that though yung lumabas na phenotype is from my father, it doesn't mean na wala na akong traits coming from my mother. I still have the genotype. Kung titingnan ang sequence ng aking DNA, meron pa rin doon coming from my mother. Hindi nga lang siya lumabas. Okay? Again, hindi nga lang siya lumabas. So, it's also the same with all the human beings. We have our phenotype. Ayun yung lumabas sa atin physically. Makikita natin like yung sa hair, sa mukha, sa body structure, sa skin color, lahat yon. Namana natin from sa mga kanunuan natin. Phenotype yun siya. Pero it doesn't mean na wala na yung ibang sequence ng genes ng like atin ng for example sa akin sa mother ko, meron pa rin yun. Andito pa rin siya sa genes ko, hindi nga lang siya lumabas. But I can also pass this on to the next generation. So for example, pag nagkaanak ako, I can still pass on the genes sa next generation. Okay? Genes coming from my mother. That's what I mean. Okay? Questions? The struggle is real. Ayoko na sa pharma lab. Bahok kayo sa katanan. Wala pa ako tana. Kaya magdiretso ko sa amino acid. Sige, sa kamate. Okay, low. Oh my gosh. Nag-loading pa yung utok. Wait lang ah. Nag-loading pa. Nagun sa demo. Busy kayo itong mga lives. I know that I am very busy, pero I know that you are busy also. You have your duty. I'll just upload the recorded video para ma-review niyo siya. Makita niyo video ako struggle. Akong now struggle din kayo karon. Okay. Let's have the amino acids. I think nag-post ko assignment kagabi eh. I just want to know kung gi-research ba ninyo siya. Let's talk about the amino acids. We started with proteins actually last time, but since we cannot produce proteins without the nucleic acids, kaya siningit natin yung discussion ng nucleic acid in between. So, balikan natin si amino acids. Nakuratan mo. Nag-ano magugo kagabi, nag-review ko para sa ako ang lesson today. Di pwede nga mag-klase nga wala nag-review, no? So, nag-review ko, wala po assignment sa ila. Simuon na ito siyang assignment. It's okay, habol nyo lang ha. Kung wala pa mo nakapasa. Sige, let's talk about amino acids. These are the building blocks of our proteins, right? Salamat sa perfect score. Ano perfect score? Perfect score sa assignment. Actually sa grades, I'm not really strict with assignments. So, ko assignment to prelim. I took midterms din. Let's see kung tama ito yung ipang-send dito ah. Anyway, this one is the amino acid structure. So, ang tawag natin, di ba, sa protein ay chon kasi meron tayong carbons, meron tayong hydrogen, we have the oxygen, and we have the nitrogen. Kaya again, we call proteins as chon. So, minsan sa exam or even sa board exam, they don't really write the word protein. Instead, they write chon there. So, do not forget, it's referring to proteins, ha? Chon. Pag cho lang siya, CHO lang, that's carbohydrates. That's the difference of the carbs and the proteins. Now, this is the general structure of our amino acids class. We have a chiral carbon at the center. So, di ba, ang ibig sabihin ng chiral carbon is a carbon bonded to 4 different functional groups. Different naman talaga sila. You have there the COOH, the carboxylic group, the hydrogen, then we have the amino group here, and on this side of the chain is the R group. So R group can be any functional group, ha, any functional group. That's the R. So pwede siya alkane, pwede siya alkyne, depende na kung ano yung nandyan. Basta any functional group yung R na yan. Now, at physiological pH, meaning our body's pH, it's between actually 7.35 to 7.45, our amino acids exist as Zweiter ions. So, nag-a-assume sila class ng charge, okay, sa body natin at physiological pH. So, anong ibig sabihin yan? Yung COOH, this one is the acid, and then this one is the base. Mag-dissociate yan sila, and ito yung kalalabasan. Magbabaliktad silang dalawa. Si COOH will just become COO. I-give up niya yung hydrogen niya. Magiging base siya. And then, yung tatanggap ng hydrogen is the amino group. So, magiging NH3 plus na siya. Mag-a-assume siya again ng positive charge. So, this will become the acid now. Ang tawag niyan, kapag mag-assume na ng charge yung ating amino acid sa body natin, is the Zweiter ions. So, that is the reason why we need to maintain a certain pH in our body, yung 7.35 to 7.45. Kasi kapag either nag-acidosis or nag-alkalosis, isa sa maapektuhan yung ating mga amino acids. Kasi the amino acids will only be functional if they are Zweiter ions. So paano kung bumaba or tumaas yung pH ng ating body, hindi na sila mag-a-assume ng charge. So hindi na sila magiging Zweiter ions and they cannot really function. So paano na yung kailangan natin na amino acids or yung proteins mismo sa body natin. Okay? So kasali ito sa board exam. You have to classify the amino acids whether they are non-polar, polar, acidic, or basic. And then paano mo siya ma-identify? Kapag mag-review kayo sa board exam, I suggest that you draw the structures of these amino acids. Kasi sometimes sinasali nila ito sa area 2. Okay? Ito naman, ito yung ating general structure. Saan sila nagkakaiba? Dito sa R. And itong R na ito will also determine whether they are non-polar, polar, acidic, or basic. So you try to look for the structures of these amino acids. Okay, again, you try to look for the structures of these amino acids and you can see there na yung mga under sa acidic, the R there, are ano siya, carboxylic acid din yung kanyang functional group. Okay? Kaya nasa acidic sila na classification. Yung mga non-polar, most of them, mga hydrocarbons yung kanilang R group. So just like glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, and the methionine. Lahat sila non-polar yung R group. Yung polar naman mostly yung may mga OH. Pag may hydroxyl sila sa side chain nila, ayun, polar yun siya. Like serine, threonine, tyrosine, ano nga ito? Aspartic acid ito. So asparagine, cysteine, and the glycine. Ano nga itong GLN? Nakalimut na ko. This is glutamic acid, aspargic acid, histidine, arginine, lysine. What is this? GLN. Glucine? No. Nakalimut ko. What is GLN? GLN, glutamic acid. GLN is, this one is glycine. Glutamine. Sorry, that's glutamine. This is glutamic acid, this is glutamine ha. Wala ko sipon. Glutamine, and then this is glutamic acid. Now, aside from that, tinatanong din nila ito sa board exam, which among the amino acids are aromatic. They are called aromatic because yung side chain nila, yung R group nila, contains a benzene ring. So, aromatic, we have the phenylalanine, tryptophan, and the tyrosine. By the way, aside from familiarizing the names and the symbols, the 3-letter symbols of these amino acids, you have to also be familiar with the 1-letter symbols nila ha. So it will be your own na research work. Pero kung gihatag na siya ni Ma'am Cherry sa inyong biochem na time, you can review your notes for this. Hanapin nyo yung 1-letter nila na symbol. Okay? So just like alanine, it's A. What is glycine? Hindi ko na ito memorize. Yung mga ano nila, 1-letter symbol nila. Ito na lang alanine yung maalala ko. The valine is V. So ayun, please review on that because baka hindi complete name or hindi 3-letter na symbol yung ibibigay sa exam. Either sa akong exams, sa inyong mga exams during the review, or sa inyong board exam mismo. Now, the sulfur-containing amino acids natin are the following: the methionine, the cysteine, and itong cis, this is the symbol or the abbreviation. Not really the abbreviation kasi mas mataas siya, but cis is called cysteine. Okay? Let me write this. Cis is cysteine, and then from cysteine, you can produce the cysteine. That's another amino acid. And then the OH containing, so most likely this will be polar. We have the serine, tyrosine, and the threonine. Okay? Please take note of that. Ito yung assignment ko sa inyo. So let me see if nag-research kayo. Ano yung not optically active na ano natin? Wala naman type.
Glycine, ma'am.
Ano? Sorry. Glycine. Let me write. Glycine. This one is glycine. I-open ko yung chat ha and look at the participation here. Binaliktad ko kasi since I'm not into giving assignments, yung aking grading actually is 40%, diba, exam, 30% pa rin yung quiz. Ang ginawa ko, 20% participation and 10% lang na assignment. So actually, nag-review ako ng sino yung nagpa-participate sa class. Syempre yun yung bibigyan ng mataas na score sa participation. RIP sa nakarens. Okay, this one is glycine. Hindi siya optically active class because if we are going to draw the structure of glycine, you have the COOH here, we have the H, and then we have the amino group here on the left, and then yung R group niya kasi class is H. So, di ba, magiging optically active lang if you have the presence of a chiral carbon. And then, what do you mean by chiral carbon? It's a carbon bonded to 4 different functional groups. This time, hindi na siya 4 different functional groups kasi magkapareho na yung hydrogen sa side chain and then this side also. So, kaya siya tinawag na, kaya siya not optically active. How about the indole group?
Tryptophan.
Okay, it's tryptophan. Yung structure ng tryptophan, yung side chain class, merong indole ring. Okay? Now, how about the precursor for the synthesis of the nicotinic acid?
Tryptophan.
Ayan sa?
Tryptophan, ma'am.
Ay, sorry, nagsulat kong niacin. It's the tryptophan.
Tryptophan.
It's the tryptophan. Tryptophan pa rin ito. From tryptophan, ayun, gagawing niacin yan. Okay? Tryptophan lang sa kalam. How about the precursor of the 5-HT? 5-HT is the 5-hydroxytryptamine. And this is class the serotonin or our happy hormones. Ganun pa rin, tryptophan. Okay? Kapoy na kong sulat sa tryptophan. TRP na lang. Tryptophan. Okay. Now, how about the precursor of cysteine? Cysteine is a sulfur-containing acid.
Serine.
It's the serine. Ano naman na yung or? Sa answer dire. It's the methionine. Kapag nakatutunlan, ayaw ka na multiple choice. It's the methionine. How about the one with the amino group? Very good. It's the proline. Okay. It's the proline. Actually, si proline can also be included here. Hindi din siya optically active. Precursor of dopamine, epinephrine, and norepinephrine. Kasi unjun tamo ani ha, naglasang yata ani sa pikol. It's a tyrosine. Do not forget that. Diba? So, naga-drawing ta sa una, o nga ni, before mabuo ato ang norepinephrine, even the epi and the dopa, tyrosine muna. That's an amino acid. Precursor of our histamine, the one that causes allergy. It's the histidine. Okay? Pwede din specific, L-histidine. Okay, but anyway, it's the histidine. And the one with the guanidine group, yung kanyang R, may guanidine group. Take note, it's the arginine. Let's see your answers. Wala na po actually nagtanaw sa answers ninyo. Arginine. Okay. Minot na akong ilong, tapos katulak akong tutudlan, ayoko na. Nagdaghan pa kong himuon. Sige. Ayan ha. So those are the answers for our amino acids. Do not forget that. Okay, so how about this? Hindi ko siya sinama, pero are you familiar with our essential amino acids? When you say essential, we cannot make it sa body. We cannot produce them sa body natin. So we need to get them from the food that we eat. Ito yung mga essential amino acids natin. Ano kaya yan? Yan yung ginagamit natin class na mnemonics ha. Private Math Hill or minsan, merong mga lecturer na private team hall yung ginagamit nila. Private team hall. But anyway, this is the same. These are the same amino acids. Yung ano lang, mnemonics lang yung nagkaiba. Private Math Hill or private team hall. Ano yung P? This one is
Phenylalanine.
syempre the phenylalanine. Isang P lang naman yung ating amino acids. And then, syempre V is the valine. How about the T? We have 2 T's only here. So, ilang T's ba yung meron natin? 3 ata. So, ano yung hindi kasali? Excuse me.
Tyrosine.
Tyrosine yung hindi kasali. So, yung nasali dyan sa essential amino acids are this one, the threonine, and what's the other one?
Tryptophan.
Okay, the tryptophan. So do not forget the exception ha. The exception there is the tyrosine. The tyrosine is not included. So definitely tyrosine is not essential. We can produce that in the body. So M is, we only have one M. Methionine. And then A. Ay, hindi siya mo agi. Siyempre. Ayan. Ilang A meron tayo? Alanine, arginine, aspartic acid. 3 yun? 3. So, isang A lang yung nasali sa essential amino acid. And that is the arginine. So, baliktad sila ng T. Sa T, 3 T, pero isa yung wala. This time sa A, 3 yung A, isa lang yung nasali. Don't forget. Ay, 4 pala. Ano yung hindi ko na? Asparagine. Asparagine pala. Isa lang nasali here, the arginine. How about the H? Ilang H tayo? Isa lang.
Histidine.
Histidine. And then the I is isoleucine. Isoleucine. And how about the L? Ilang L tayo? 2 lang din, no?
Lysine.
Automatic kasali silang 2. Lysine and the leucine. So do not forget, you have to be familiar also with all our essential amino acids. Anong ibig sabihin ng essential? We are not able to produce this. And ang mangyayari, kailangan natin siya i-consume from the food that we eat. Okay? Sige. Please take note of that. If hindi niyo siya mahabol, anyway, this one is being recorded. Okay, next. Actually, na siya sa next slide. Okay. And then ito yung mga non-essential natin. Mga conditionally non-essential, and then we have here the non-essential. We have alanine, aspartate. This is our aspartate, not aspartate. Aspartate, cysteine, and glutamate. Basig malibog mo kasi si arginine is like included here supposedly in the essential pa, but it can also be considered conditionally non-essential depending on the condition of the patient. Okay? So we have asparagine, glutamine, glycine, proline, serine, and tyrosine. And then the non-essentials are this one. Okay? Now another thing that you have to know is if the amino acids are glucogenic, ketogenic, or both. So yung all the others class are actually glucogenic. When you say glucogenic, they are metabolized into, anong tawag dyan? Nag-undergo sila ng gluconeogenesis. Ano ibig sabihin ng gluconeogenesis? It's making a sugar out from non-sugar sources. So it can be proteins class. And the amino acids that can be converted into sugar are called glucogenic. Okay? So all the others na wala dyan sa listahan are glucogenic. So pwede na sila mag-undergo ng gluconeogenesis in times na kailangan natin ng sugar tas wala na tayong other sources. Now, pag ketogenic naman, sila yung mag-undergo ng ketogenesis to form ketone bodies. Or mag-una, i-metabolize sila to make the acetyl coenzyme A that is needed for like synthesis sa fats or sa lipids. And then ang end product will be the ketone bodies. So we only have two, leucine and lysine. And for both na pwedeng mag-undergo ng gluconeogenesis or mag-undergo ng ketogenesis, we have the PIT. So we have phenylalanine, isoleucine, tryptophan, and tyrosine. All the other amino acids not mentioned here are glucogenic. Okay? Questions? Ang oras na. Hanggang 6 tayo na. We'll take a break at 4:30 para hilipod kay ta bombarded. Wala pang tana. Okay, now let's talk about the structure or the structural organizations of our proteins. We have classed the first one, we are talking about proteins. Primary structure of the proteins is just the amino acid sequence. So kapag yung sa video kanina na idugtong-dugtong lang yung mga amino acid na yan, ayan yung amino acid sequence. Primary, yan yung primary structure natin for our protein. So it's just a polypeptide. Now, itong mga amino acids na ito yung ating bonded together by the so-called peptide bond, which is a covalent bond. So it's a strong bond. That is why hindi basta-bastang nade-destroy si peptide bond unless merong enzymes talaga intended for that. Covalent bond siya. Wait, catching ko sa Okay, sorry for the interruption. Okay, let us continue. So again, peptide bonds are covalent bonds, and this is where we can find the disulfide bonds also if mag-form na ng ibang structure yung protein. And this is a partial double bond in character. Partial only kasi you can only see the bonding sa COOH na part. So, hindi the whole structure. So, partial double bond in character. It's an amide bond. This is a board exam question. Amide linkage ito. Paano siya naging amide? Wait. Ayan. So, di ba yung ating general formula for our amino acid looks like this. This one is an R. So, di ba you have the C double bond O here, and then supposedly this is OH kasi COOH siya, di ba? Ang mangyayari, pag mag-bond na siya with another amino acid, matatanggal yung hydrogen dyan, and then, dudugtong siya ngayon dito sa another amino acid. So, this one class is the peptide bond here. Anong nandito? It's the N. Pero hindi na siya NH2, NH na lang siya. Makukunan siya ng isa. And then magpapatuloy yung structure. So for example, dalawang amino acid lang. So again, this one is the so-called peptide bond here. It's partial double bond in character because of the presence of this one. And it's an amide linkage. Ibig sabihin ng amide, di ba? RCO and H2. So RCO and then you have the NH. So this one here again is an amide linkage. Again, this is a covalent bond. This is not easily destroyed except if you have enzymes. Hindi siya madadenature kaagad. Okay? So this is the primary structure of our proteins. Ano lang yan siya, mga bilog-bilog dyan, amino acids lang yan. Now, how about the secondary structure? This one is formed if you have like amino acid that are close to each other. So, linear sequence pa rin siya. Ito pa rin siya, ganito. Pero because they are close to each other, magfo-fold yung protein and magkakaroon ng bonding. Actually, the bonding between the foldings are hydrogen bonds. So ito yung nadagdag aside from the peptide bonds. If the primary sequence folds, ayun, may H bonds in between na mafo-form. So anong examples ng ating secondary structure for the proteins? We have the regular, just like the alpha helix and the beta pleated sheets ng fibrin and ng keratin. So keratin class is an example of a secondary structure na protein. And then we have irregular na mga random coils. So wala silang definite structure. Ito, meron silang definite na folding or yung shape nila. Ito random coils lang. Example, the collagen. Take note, that is the most abundant protein class, the collagen. The most abundant protein sa blood is the albumin. Okay? Pero kung as a whole, that is the collagen. Now again, this is the secondary structure. I'll show you a picture later. And then we have the tertiary structure of the protein, which is now a 3D structure. So you have now 3 sides, 3 dimensions, and then you have the domains for the tertiary structure. And aside from the peptide bond and the hydrogen bonds, let's add more bonds to this. We have the disulfide, hydrophobic, and the ionic interactions or ionic bonds. So examples of our tertiary structure are the globular proteins and the fibrous proteins. Pag globular, syempre yung bilog, spherical in shape, and they are water soluble, just like our hemoglobin and our myoglobin. Both are carriers of oxygen, found in the red blood cells kung sa blood, and then sa muscles natin yung myoglobin. And then the fibrous one, fiber. So they are elongated and they are insoluble to water. Samples, we have the collagen, the elastin, and the fibrin. Kasama si collagen dyan. And then we have the quaternary structure in which we have polypeptides. Maraming polypeptides na nagdikit-dikit with each other, nagdugtong-dugtong. So again, pag quaternary na, meron na tayong multi-chain na protein here na nagdugtong-dugtong. So hindi lang siya isang peptide, so polypeptide na. We have additional bonds such as the hydrogen, hydrophobic, and ionic interactions here. So to compare, ito yung primary structure na nagdugtong-dugtong lang yung ating amino acids. And then ito yung secondary structure kapag kayong primary structure is nag-fold na, and the folding happens because of the presence of the hydrogen bonds. And then ito yung tertiary structure, so hindi lang hanggang folding. This one is a complicated structure na it can form a sphere like the globular proteins or an elongated strand like the fibrous proteins, pero maraming bonds na yung involved dito. Kasi maraming folding na yun, a complicated folding na. So tertiary structure na yan siya. But if you have quaternary structure, take note, marami ka ng polypeptide chain na nagkumpul-kumpul, nag-bond with each other. So may mga additional bonds din involved dito. Ito na yung quaternary structure. So take note, class, that secondary, tertiary, and quaternary structure can be denatured easily. Even just by heating, heating, the presence of metals, but the denature na kaagad yung secondary, tertiary, and quaternary structures. But the primary structures cannot be easily destroyed except if we have enzymes available para ma-destroy siya. Okay? So again, ang explanation dyan kasi the peptide bonds between the amino acids are covalent bonds. They are considered strong bonds. Okay, questions so far? Wala pangutana. So, take note of this. Protein metabolism starts in the stomach because this is where we see the enzymes involved for metabolism. And we call them peptidases. Okay? Peptidases. So these are divided into two. Exopeptidases if they will split the carbon and the nitrogen terminal of the amino acid. So, di ba meron kang nitrogen on the left side, for example, and then the carboxylic acid on the right side. If split yan sila, yan yung exopeptidases. So example, the carboxypeptidases and the aminopeptidases. But if the metabolism really is in the yung nasa gitna talaga ng ating structure that's within, so endopeptidases yan. Example, the pepsin and the trypsin na sisirain niya talaga yung peptide bonds. So sa gitna. Again, we have the pepsin and the trypsin. Pepsin is found in the stomach. That is the reason why protein metabolism starts in the stomach. Okay, take note of that. Now, I'll just, kapag nasa enzymes na tayo, mag-break muna. Mag-break tayo dyan pagdating sa enzymes. Tapusin ko na lang itong protein metabolism condition. So ito yung mga sakit involving the problems in the metabolism of protein. So the first one is the PKU. If you hear this condition, that is called phenylketonuria. And one symptom or one sign of the PKU is yung urine ng tao is kaparehan ng amoy ng urine ng daga. So yung mga nag-experiment sa school with the research or yung nag-alaga ng daga sa PICOL, you have smelled, class, the urine of the mouse, and if ang urine ng person, class, magiging the same yung amoy ng urine sa daga, pwedeng meron siyang phenylketonuria. Now, what happens if a person has phenylketonuria? There will be an increase in phenylalanine and a decrease in tyrosine. Again, phenylketonuria ang tawag. The reason is there is lack of the enzyme phenylalanine hydroxylase. So isang pathway actually yan sila ng alkaptonuria, which is the second condition. So in the body kasi, class, if you have phenylalanine, through the enzyme phenylalanine hydroxylase, it will be converted into tyrosine, tapos may mga other enzymes involved pa dyan hanggang maging mallyl acetoacetic acid siya. That's how we metabolize the phenylalanine. Now, our problem with PKU is the person lacks phenylalanine hydroxylase. So, bababa ngayon yung tyrosine, hindi mako-convert si phenylalanine to tyrosine, and then ito, syempre, tataas. So, ang ending, magiging kagaya sa daga, yung amoy ng urine. Okay? And then, the second one is the alkaptonuria. Ito naman, there will be darkening of the urine. So, magiging very brown, as in dark ang color ng urine. And there will be earwax upon standing. What do you mean by earwax upon standing? Titigas yung urine, class. So nakakita naman tag-urine nga hindi natapon, example sa Arenola, hindi natapon overnight. Murag, kung sa layman's term, pamapanos siya, baho na kayo siya, pero hindi siya tumitigas. Pero pag ang person may sakit, called alkaptonuria, upon standing, class, titigas yung ihi nila. So aside sa different ang color. So what happened in a person with alkaptonuria, there is an increase in tyrosine, and at the same time all the other homogentisate derivatives, and the problem is on the enzyme called homogentisate oxidase. So ipagpatuloy lang natin ito, si phenylalanine will become tyrosine through this enzyme, tyrosine aminotransferase. We have here 4-hydroxyphenylpyruvic acid. We have another enzyme. And then, ang next na product niya is homogentisic acids. Ito yung derivative. Now, ang problema for alkaptonuria is this one. Walang enzyme na to. Homogentisate 1,2-dioxygenase. So, syempre, tataas yan. Tataas ito. Tataas si tyrosine, even the phenylalanine. Kaya ang ending, magda-dark yung urine and titigas siya upon standing. So, yan yung difference na ngayon ng phenylketonuria and the alkaptonuria. Okay, please be familiar with this pathway. Now, how about the albinism? People that are classified as albino actually has a problem sa protein metabolism. So there is decreased melanin, class, yung pigment, kasi may problem din sila with the enzyme called tyrosine hydroxylase. So hindi ma-metabolize yung mga amino acids like tyrosine to form melanin. I mean, metabolism is both anabolism and catabolism, no? So, hindi ma-form si melanin, walang process, yung pathway hindi matutuloy para ma-produce yung melanin na yan. And then again, the enzyme that is lacking sa mga persons na albino is the so-called tyrosine hydroxylase. So, sa proteins yan sila, may problema. I believe you saw already a person that is albino. Anong puti kayo sila, pati ilang buhok, pati ilang kilay, pati ilang tanan, puti. Okay? So protein metabolism yung problem actually sa kanila. And then meron tayong tinatawag na MSUS or the maple syrup urine syndrome. So ang ilahang urine, murag maple syrup ka nang ginabutang sa pancake. Okay, kanang ginabutang sa pancake na lapot, and at the same time brownish in color from the maple leaves. So mo na nga maple syrup urine syndrome ang tawag. that is still a problem in protein metabolism. There will be increase in the LIV. LIV na amino acids, leucine, isoleucine, and the valine, because hindi na sila ito tatlo hindi ma-metabolize due to the deficiency in the enzyme involved in the metabolism of these amino acids, the so-called alpha keto acid dehydrogenase. So kalayan na mangihi kano nalapot ang imuhang ihi. So this one is an example, a picture of a person that is an albino. So, kulang sila ng melanin kasi kulang sila ng enzyme that will metabolize tyrosine para again ma-form si melanin class, which is a pigment sa skin, sa tanan, sa buhok nato. Okay? Questions so far? And then lastly, we have the homocystinuria. Another problem in protein metabolism, but this time may malaking problem tayo dito because the person with homocystinuria has mental retardation. Mangitom. They are very sensitive to sunlight. Dali sila magsakit. They are very prone to like skin cancer since they don't have protection from sunlight. Di sila mangitom, magpula guro sila. Pero sakit kayo na para sa ilahang sunlight. Labi na sa ilahang mata. Okay. Going back to homocystinuria, the person has mental retardation, osteoporosis, and lens dislocation. So what's the problem? There is increased methionine and homocysteine because of the deficiency in the enzyme cystathione synthetase. So, di ba nasabi na kasama sa assignment ninyo para ma-produce si cysteine, galing siya kay methionine. Pero since there is an enzyme that is lacking here, methionine and homocysteine lang siya, hanggang doon lang siya. So yun yung mag-i-increase, and then again, the person will have the condition homocystinuria. And ito yung makikita natin sa persons na may homocystinuria. And that may be present at birth. Okay? So kaya may mental retardation. So ito yung mga sakit again involved in problems with protein metabolism. So, mga enzymes ang problema niya. Now, we will take a break and then afterwards, we'll talk about enzymes. 20 minutes na break, so be back here at 4:59. Okay, recording. Okay, let's talk about the enzymes class. So since this one is a review class, I believe you are very much familiar with the enzymes. These are our biological catalysts used to speed up reactions that take place within our body. So they are lowering the energy of activation. That's how they speed up the chemical reactions taking place in our body. Now, we have different types of enzymes. The first one is the zymogen or our proenzyme. When you say zymogen, these are inactive enzymes. They are yet to be activated. Examples of our zymogen, we have the pepsinogen. Pepsinogen yung inactive, yung zymogen, na kapag in the presence of hydrochloric acid, they will be activated into pepsin. So the pepsin now is the active form or the complete form of the enzyme, or that is what we call the holoenzyme. Pag inactive, zymogen pa. Pag active, holoenzyme. So another example, we have the trypsinogen, which is the zymogen or the inactive form. And through the presence of another enzyme called enterokinase, trypsinogen will be activated into trypsin. So both pepsin and trypsin are called holoenzyme or the complete active enzymes. Okay, mga active enzymes, yan sila ng mga counterparts nila ng mga zymogen. Now, That's the two types of the enzymes. Now for the parts of the enzymes, we have the protein portion or the protein part, which is called the apoenzyme, and the non-protein portion is called the cofactor. Now we have two types of cofactors. The first one is the prosthetic group, which is an inorganic substance. So any inorganic substance that is attached to the apoenzyme to complete the whole enzyme is called the prosthetic group. And they are covalently bonded to the enzyme, or they are firmly bound to the enzyme class, to the apoenzyme. But if the cofactor or the non-protein portion of the enzyme is an organic substance just like our vitamins, ang tawag dyan ay coenzyme. Okay? So, again, pag mga organic substance, coenzyme ang tawag. Kapag inorganic, just like our metals, di ba? Our metals, labi ng mga trace metals na to, important sila as cofactors for our enzyme. Ang tawag natin sa kanila ay prosthetic group. Okay? Now, we have 6 major enzymes class. This is depending on their work sa body or sa chemical reaction. So we have here the isomerases which catalyze the isomerization reaction. So if you want, di ba, when you say isomer, these are substances with the same molecular formula but different structural formula. So if you are going to convert one substance to another isomer, the process is called isomerization, and then the enzyme that will catalyze the reaction is called the isomerase. Okay, marami yan sila, so kaya tinawag siyang isomerases. And then another one, we have the hydrolases. From the name itself, hydro, this involves water. So what happened? There is removal of the functional groups by the addition of water. So ang kinakatalyze na reaction is syempre hydrolysis. Okay? One example of an enzyme that is under the hydrolase is the amylase. So di ba the amylase, like our salivary amylase class, if magkaroon siya ng interaction with food, it will actually remove some functional groups from the food that we eat through the water. Okay? Through water pa rin itong action ng ating amylase or our salivary amylase. That is also true for all the other hydrolases that we have. Now, for the transferases, from the name transferases, this will catalyze the transfer of some important atoms or elements in our body. So like transfer of carbon, nitrogen, or phosphorus containing groups. So ito yung mga enzymes na nag-e-end sa kinase. Okay, like phosphokinases or any other kinases that we have that will just catalyze the transfer of these specific atoms or elements. Next, we have the lyases. For the lyases, they will cleave the carbon to carbon or carbon to sulfur or carbon to nitrogen bond. So pag hihiwalayin, for example, we have chemicals na that is made up of carbon and another carbon. Kung gusto nating ihiwalay yan, lyases yung tawag sa enzyme that will catalyze the cleavage. So examples of our lyases, we have the decarboxylase and the deaminases. From the name itself, you can predict there kung ano yung kinikleave nila or ano yung sineseparate nila. Siyempre, the carboxylase will cleave carbon to carbon bonding and deaminases will cleave carbon to nitrogen bonding. Next, we have the ligases. Baliktad, if si ligase will cleave or separate the atoms, the ligase naman will join them together. Okay, so mag-coupling ang two molecules. So kagaya ng trabaho ni DNA ligase in the DNA replication. So remember the Okazaki fragments of the lagging strand. Magkahiwalay-hiwalay yon. So in order for us to join them together, we need the DNA ligase. So under yan siya sa mga ligases group of enzymes. So another example is yung carboxylase natin. Nagdudugtong din siya ng mga molecules. And syempre, oxidoreductases, if the enzyme catalyzes redox reactions like dehydrogenases and oxidases na mga enzymes. Again, involved sila in catalyzing redox reactions. Okay, questions? That's all for our enzymes. Anything that you want to clarify? Wale, sure na. Sure na, Jud. Okay, we will be talking about carbohydrates, but hindi tayo masyadong mag-dwell with the carbohydrates since we are done with this sa organic chemistry. So dadaanan lang natin ulit yung carbs natin ha. So again, yung ating general formula for carbohydrates is CHO. Carbon, hydrogen, and oxygen. And then so protein, not the general formula, but the abbreviation. And for proteins, we have the CHON. Now take note class that our carbohydrates can either be aldehyde or ketone. So pwede siyang aldose, pwede din siyang ketose. Now what is the building block of the carbohydrates? We have the monosaccharides. If it is amino acid for the proteins, nucleotides for the nucleic acids, we have the monosaccharides for our carbohydrates. Okay? Now, kung amino acids are joined together by peptide bonds, which are amide bonds, this time naman, monosaccharides are joined together by glycosidic bond. Take note that glycosidic bond is an ether na bond. Not the ether. I don't know. Yeah, that's ether. ROR. So, oxygen lang nasa gitna. Okay? So pag sinabing monosaccharide, it's one sugar unit. Siyempre pag dalawa, di. Pag tatlo, tri. Actually pag 3 or more, that's oligosaccharide. But if it's more than 10, that's the time we call it polysaccharide. So take note that mono and disaccharides are sweet except our lactose. Lactose are actually not sweet. But all the rest, all the other mono and disaccharides are sweet. And then, just for differentiation between the mono and the polysaccharide, we have here for the solubility in water, monosaccharides are soluble in water. Even the disaccharides, like our sucrose, ang yung asukal natin, can be dissolved in water. So that is why maraming plus yan. But the polysaccharides are not soluble in water. You have tried that when you made, I don't know if you have made a starch paste. Yung starch mismo na powder, di ba? Dili siya ma-dissolve sa tubig. Instead, mag-form lang siya ng paste. Okay? And then, crystallinity, monosaccharides are they exist as crystals, so they have definite lattice form or lattice arrangement, while the polysaccharides are amorphous. So you can differentiate that one if you have glucose, fructose, or even our disaccharides like the sucrose, naka-crystal talaga, and then yung starch, hindi siya crystal, diba? It's just a fine powder, so amorphous siya. For the sweetness, I have mentioned this, both mono and disaccharides are sweet, but polysaccharides are not. Hydrolysis, since at ito na yung pinaka-last building block na talaga siya ng carbohydrate, so hindi na siya mag-undergo ng hydrolysis. But polysaccharides, being a very big molecule, can undergo hydrolysis. But both of them have optical activities due to the presence of our chiral carbons in the structure. So I think this table is also included in our discussion sa organic chemistry. The names of the carbohydrates depending on the number of carbons and depending on the functional group that is present. Since carbohydrates can either be a ketose or an aldose, so yung depende kung ano yung nag-exist, aldehyde ba or ketone. So please be familiar with the names of the carbohydrates, just like when you have a 5-carbon na aldose, we call that pentose. But if it is a 5-carbon na sugar, tapos ketone yung functional group na present, we call that pentulose. Okay? And then specific example, ito kasi yung general name pentose kung 5 ka sugar. Ang specific example, the ribose that is found in our RNA is actually an aldose. 5 carbon, tapos aldehyde yung kanyang functional group. And then the most abundant and the most important carbohydrates are actually the hexose. Kasi board exam question yun ha, hexose yung ating most abundant and most important carbohydrates. Hexose yun, 6 carbon sugar, and at the same time, most of them are aldehydes or aldose. Just like the glucose and the galactose. Though fructose are also important since these are our fruit sugars, pero hexulose kasi siya, ketone yung functional groups. Now, be familiar with the different structures of our carbohydrates. If it looks like this class, this is what we call the Fisher projection. Okay? Itong linear lang class na structure ng sugar natin, ang tawag natin dyan, Fisher projection. So ito yung Fisher projection for some of our aldotetroses. Okay? Tetrose kasi 4 na carbon, aldo because these are all aldehydes. So etong carbon number 1 is an aldehyde, CHO. So we have our CHO. And then if it looks like a ring, just like this one, ito yung tinatawag na Haworth ring. Okay, Haworth ring ito. And pwede din natin gamitin actually si B and si L and si B dito. Depending yun siya kung saan makikita yung OH. Yung OH yung tinitingnan dito whether nasa left or nasa right. But pag sinabing L, syempre sa left, D is para sa right. Okay? We'll talk about that later kung saan ilolocate yung left and right na yan. Okay? Now, aside from the Fisher and the Haworth projection, we can also elucidate the structure using the chair conformation and the boat conformation. So chair, it really looks like a chair and is actually the more exact structure of our carbohydrates. It will give us the 3D structure of our sugars. But when it comes to stability, the more stable structure for our carbohydrate is actually the boat conformation. So these are the comparison. We have here the Haworth projection wherein this one is actually the more common one, more common projection. This is the chair conformation. So it looks like a chair. That is why it's called chair conformation. It's the more exact structure for our sugars. And this one is the so-called stable structure, the boat conformation. It is also called boat conformation because it looks like a boat. Okay, to be familiar. And then both Fisher and Haworth projections are actually commonly used. So this one now is plus a Fisher projection, and we will just differentiate a ketose from an aldose. So si glucose plus is an aldose because it has aldehyde as the functional group. You may look at the carbon number 1. So this is R, CHO here. So glucose again is an aldose. But fructose is a ketose because it has a ketone as its functional group for the terminal carbon. So this is R. R is any functional group. R, C, double bond O, and this is considered an R, which is the general formula for a ketone. Okay, that's how you differentiate an aldose from a ketose. Oh my gosh. Okay, so ito na yung differentiation between the D and the L. Paano natin tatawagin na D? Paano natin tatawagin na L? Itong D and L ng ating carbohydrate is not actually referring to the optical activity. Diba, optical activity refers to the ability of the substance to rotate a plane polarized light either to the right or to the left. So if it's to the right, it's dextrorotatory. If it is to the left, it's levorotatory. But for carbohydrates class, the D and the L are actually referring to the position of the hydroxyl group of the carbon which is nearest to the last carbon group. So just like this one, this is carbon number 1 here. the functional group for the aldehyde or the ketone are group number 1 or carbon number 1, and then this is the terminal here. This is the last carbon, and then the carbon before the last carbon class, ito yung tinitingnan natin to know whether the substance is a D or an L. So both of this, D-glucose and D-fructose, both of these one, glucose and fructose are actually D. Bakit? Kasi yung second to the last carbon, yung kanilang OH nasa right side. Okay? Pag nasa left side na yan, ayan yung tatawagin nating either L-glucose or L-fructose. I hope that is clear ha for carbohydrates. The D and the L are not referring to the optical activity. It's referring to the position of the OH sa second to the last carbon. Okay? And then we have this one sa ating organic chemistry, the epimers under the isomers class. So nag-exist ito sa mga carbohydrates. Pag sinabing epimers, they have the same molecular formula. Again, the same molecular formula, pero different yung kanilang structure. For epimers, they differ only in the configuration at one asymmetric carbon. Magkapareho na silang lahat except sa isang carbon. That's epimers. So you learn this one sa organic chemistry. The D-mannose and the D-glucose are epimers at carbon number 2. So they only differ at carbon number 2 because the position of the OH and the H is nagkabaliktad, nag-exchange. While D-glucose and D-galactose are epimers at carbon number 4. So again, magkaparehas lahat ng carbon dyan except sa carbon number 4. Okay, I hope that is clear. Now for oxidation, for oxidation, this is just for glucose. Again, carbon number 1 is for the aldehyde na part ha, and then carbon number 6 itong CH2OH. Now, there are parts of this structure that is viable for oxidation. It's actually the carbon number 1 and carbon number 6. If itong aldehyde, remember class, si aldehyde kapag mag-oxidize siya, pwede siyang mag-form ng carboxylic acid. Okay? And then itong sa baba, which is an alcohol, ganun pa rin. Pag mag-oxidize, pwedeng maging aldehyde, and then yung end part niya, maging carboxylic acid pa rin. So kaya kung titingnan ninyo, acid yung kalalabasan if either carbon number 1, carbon number 6, or both carbons ang ma-oxidize. So si carbon number 1 for glucose, this one, if ma-oxidize siya, magiging acid siya, COOH. Instead of glucose, tatawagin natin siyang gluconic acid. Okay, that is actually an aldonic acid. Pero if parehas lahat ang na-oxidize itong carbon number 6 maging COOH ito, it's a uronic acid. For glucose, we call that glucuronic acid. So ito yung sa glucuronidation natin sa metabolism na part. It's actually a glucose wherein the carbon number 6 has been oxidized. But if both carbons will be oxidized into COOH, the glucose will be called glucaric acid. Okay, glucaric acid. Now, itong pag-oxidize sa galactose, this one, if both carbon number 1 and carbon number 6 ma-oxidize, ang tawag naman natin sa kanya galactaric acid or the so-called mucic acid, in which ginagamit natin siya as a test for identification of the galactose, the mucic acid test. Okay, ipapa-oxidize natin. Example, we have a sample and we suspect that it is a galactose. We will just make both carbons, carbon number 1 and number 2 oxidized, and then a positive result will give us a white precipitate. Again, that's called mucic acid test for galactose. The white precipitate there is called the galactaric acid. Okay? Now, that is for oxidation, but our carbohydrates are also capable of being reduced. Okay? Mari-reduce din sila. Now, if our sugars are being reduced, instead of carboxylic acid, ang ipoproduce nila alcohol. So, pabaliktad tayo ha. Pabaliktad tayo. Nasa gitna si aldehyde kasi. So, si aldehyde magiging as carboxylic acid if ma-oxidize and magiging alcohol kapag na-reduce. So ito yung tinatawag natin na sugar alcohol. So just like glucose, if it undergoes reduction, it will give us the so-called sorbitol. Other name for sorbitol is the glucitol. Actually, mannitol is also considered a sugar alcohol. Okay? So mannitol plus is used as a diuretic, but it's actually now used more on the pag-decrease sa intracranial pressure. Mostly pag nag-work kayo sa hospital, kapag ka may mga brain tumor or may brain injury ang patient, binibigyan talaga ng mannitol. Because of its osmotic action, kukunin niya yung tubig sa ulo. Okay? Okay? We have here the anomers. This is another form of isomer, but this time the difference is on the carbon number 1. Baka malito kayo ha, doon nag-lesson na tayo nito sa organic chem. Si epimer, any carbon. Mag-differ sila sa any carbon as long as isa lang. Si anomer, ano talaga siya, specific at carbon number 1. And we can see this class kapag ka naka-Haworth projection yung ating sugar. Okay? Meaning naka-ring siya. Okay? Again, si anomers can be seen if the sugar is in its Haworth projection. So again, we will just look at carbon number 1. If the OH for carbon number 1 is found below the ring, it's called an alpha anomer. So kung makikita nyo, meron tayong tinatawag na alpha D glucose. Meron din tayong tinatawag na beta D glucose. Okay? So alpha and beta is actually referring into the position of the OH of the carbon number 1. But if it is above the ring, it is called a B anomer. So both of them are actually glucose. The position lang yung nagkaiba. Okay? Any question so far? Or clarification? Kaya daghan-daghan na ni. Kaya pa? Sudyo ning biochem ba? Loading na ng uban din. Ubasig natulog na ang uban. Okay. It will just take time, so you need to study after ha. Kailangan ninyo ni siya tagaan og panahon jud. So especially if mag-take na mo og board exam, sulod ninyo tanan silang brain cells. Tanan. Okay. So we have here, we are done with the monosaccharides. Let's move on sa disaccharides natin. Di means two. So we have two monosaccharides linked together. Now most of our disaccharides are reducing sugars or reducing agents, meaning they can cause reduction to other substance, but they themselves will be oxidized. So that is except sucrose. Okay? Kasi si sucrose wala siyang free carbonyl carbon. Yun kasi yung ma-oxidize supposedly. And yun yung magkakos ng reduction ng another substance. So hindi free kay sucrose, kaya hindi siya reducing agent. So do not forget the Benedict's test for the reducing sugar. We use copper sulfate for that. And the positive result will give us a brick red color. Okay? So sucrose, since disaccharide, I believe you are very much familiar with this. Glucose plus fructose, yan. Lactose, glucose plus galactose. And if it is both glucose, that is maltose. So ito yung illustration of the sucrose. Wala siyang free carbonyl carbon kasi nagamit siya in the bonding. Okay, hindi siya reducing sugar. Now for oligosaccharides, we have more than 2 monosaccharides here linked together. Examples, we have the maltotriose, which consists of 3 glucose units. And then the dextrine, oligosaccharide din yan siya. Dextrine actually is a product of the digestion of starch in the mouth. So remember, we have the salivary amylase. And then if there is an interaction with the starch that we eat, the starch will be broken down into oligosaccharides, specifically the dextrine. And then we have the polysaccharides that are long polymer ng monosaccharides natin. So most likely it's 10 or more the monosaccharides joined together. But these are divided into two, either homopolysaccharide or hetero. Homo means we only have one type of monosaccharide in the structures. So example, we have the glucans. Gaano man kahaba or gaano man kalaki ang structure nila, as long as it's all glucose, we call that glucans. That's under homopolysaccharides. So examples of our glucans, we have the starch, which are the storage polysaccharides in plants, the glycogen, the storage polysaccharides ng tao, or even the animals, and then the cellulose, which is the structural polysaccharide of plants, and the chitin for the structural polysaccharides ng ating crustaceans. Okay? So, let's start with starch. Review na lang manina. Agian na lang nato kay humanata sa atong orgam ani. So, two components of our starch, the amylose and the amylopectin. Take note, ang structure nila, ang difference, amylose yung linear, and ano lang siya, puro lang siya glucose na linear linked together by alpha-1,4 glycosidic band. While si amylopectin yung branch, syempre kung branch na siya, dalawang klaseng glycosidic band na yon. Yung linear na part, alpha-1,4 pa rin, pero yung branching niya is alpha-1,6 glycosidic band. So the branching for amylopectin occurs every 24 to 30 glucose units. So medyo malayo pa yung branching, 24 to 30 glucose units, but take note that amylopectin is water soluble. Okay? Now for glycogen, again storage ito ng ating animals. Similar siya sa amylopectin, it's still branch and at the same time water soluble. And parehas lang sila ng amylopectin ng glycosidic band, alpha-1,4 and alpha-1,6. What's the difference? It's actually more branched than the amylopectin kasi the branching occurs every 8 to 12 glucose units. Yung isa malayo pa, 24 to 30. Okay? So again, we have amylases that are capable of digesting or metabolizing these polysaccharides. Okay? The salivary amylase is capable of destroying the alpha-1,4 glycosidic band, while the amylopsin, yung isa pa natin na amylase is hindi yung isang amylase, yung dextrinase natin and then yung alpha glucosidase natin, yun ang capable of destroying this alpha-1,6 glycosidic linkage. Okay, and then we have the chitin, which is the exoskeleton of our insects and the crustaceans. Ano din siya, glucan din siya, pero it's specific na repeating units of the N-acetyl glucosamine. Yun yung nasa, yung matigas na bagay sa matigas na exoskeleton ng ating insects and crustaceans. Okay? Now, that's all for the homopolysaccharide. For our heteropolysaccharides, we have more than one type of monosaccharides here. More than one type of monosaccharides. We have first the hyaluronic acid, which is present dun sa water ng ating mata and ng ating synovial fluid. So actually, sugar din yung water natin, the vitreous humor and the synovial fluid between our joints. So they are used class as lubricants. The name comes from hyalos meaning glass. Parang glass kasi siya kung yung appearance niya. And then we have the chondroitin sulfate. Chondroitin from the word chondrus, which means cartilage. Polysaccharide pa rin yan, present in our cartilages, tendons, and ligaments. So di ba meron tayong food supplement meron tayong preparation for arthritis na merong chondroitin sulfate. So ayan yun siya, polysaccharide pa rin yun na para sa mga cartilages, tendons, and ligaments natin. And then we also have polysaccharides sa nails and sa skin natin. Kung sa nails, we call that kermatan. And then sa skin, we have the dermatan yan siya, dermatan sulfate. And then take note class that the heparin is also a polysaccharide. So, di ba, our heparin is the injectable natin na anticoagulant to prevent coagulation of the blood. And then we also have a polysaccharide found in our seaweeds. We call that agarose. Okay? And then for our bacteria, especially in their cell wall, and specifically for the gram-positive bacteria, we have the peptidoglycan. It's a heteropolysaccharide class. Okay? So we have there a protein plus the carbohydrate or a heteropolysaccharide, kaya tinawag siyang peptidoglycan. So it's actually a combination of N-acetylglucosamine and N-acetylmuramic acid. And ginagamit yan siya to differentiate gram-positive and gram-negative kasi mas madami yung peptidoglycan ng mga gram-positive bacteria natin. Okay? And then, other things na other substances in the body na merong carbohydrates, our peripheral proteins, most of them are glycosylated. So, di ba na-mention kanina, after the ribosome synthesized a protein, it will be stored in Golgi apparatus na mag-undergo ng modification. And one modification na i-undergo niya is glycosylation. The protein will be attached to a carbohydrate. So may mga carbohydrates ng mga peripheral proteins natin. And then, take note of this, our blood group antigens are actually also made up of carbohydrates. Okay? They are actually proteins na glycosylated. Yung nagde-differentiate ng blood type, A, B, O, blood type A, B, or O, A, B, ayun, carbohydrates pa rin yan. Mga antigen, blood antigens natin. Okay? Now, if sa proteins may metabolism starting from the mouth, starting from the stomach, yung carbohydrates natin will start sa mouth. So meron tayong carbohydrate metabolism. Let's just have a review of the different processes. Pag sinabing metabolism, lahat na yun ha, including anabolism or catabolism. Metabolism yun ang tawag natin. So pag sinabing anabolism, that's building up, meaning you are going to make a larger molecule from a smaller one. Okay? Just like when you have monosaccharides linked together by glycosidic bonds forming a carbohydrate, a polysaccharide, anabolism ang tawag doon. So it needs energy. It's called endergonic process. Kapag catabolism naman, baliktad, it's breaking down. So like if you have a sugar, pag i-break down mo yan siya, kagaya ng glucose, pag i-break down natin siya through glycolysis,
catabolism ang tawag doon. Hanggang maging pyruvic acid siya just to produce the ATPs, a source of our energy. Pero pag merong process class na you can see both anabolism and catabolism, ang tawag dyan amphibolism. So just like the Krebs cycle or the TCA, the tricarboxylic acid, wherein we can get a lot of ATPs, a source of our energy. Actually, hindi siya either anabolism, hindi siya anabolism, hindi lang din siya catabolism. Pero both of them, nag-exist yan siya sa Krebs cycle mismo. So merong i-breakdown, meron ding i-build up. Amphibolism. Okay? Questions? Wala. Sige. Sure na. Ay, balikan na ninyo inyong lesson kay Ma'am Cherry nga perting taasa. So we have the glycogenesis, genesis, meaning we are making glycogen. So again, glycogenesis is the formation of glycogen. Saan siya galing? Sa glucose. The main enzyme involved in the production of glycogen is the glycogen synthase. And this is being controlled by insulin. Okay? Yung sobra-sobrang glucose from the blood will be used and mag-undergo sila ng glycogenesis, iso-store sila sa liver as glycogen. So not just in the liver we also store glycogen in our skeletal muscles. But if it's the other way around, if you lack glucose, the glycogen store will be destroyed, and the process is called glycogenolysis. Baliktad ito. So ano ito? Si glycogenolysis most likely catabolism ito. Breaking down. Glycogen will be broken down into glucose. The main enzyme involved is glycogen phosphorylase. And this time naman, it is controlled by the hormone glucagon. So si glucagon yan, kapag kakulang ka na ng sugar class sa body, si glucagon na yung at work. Magti-trigger ng glycogenolysis. Now, if your glycogen store is already depleted, wala ka ng ibang makukuhanan ng glucose and then you need energy, that's the time gluconeogenesis will happen. Okay, so we will be forming glucose from other non-sugar sources. That's the time our body will break down fats, and the fats will be converted into glucose just for us to have energy kapag nag-diet ka, tapos konti lang ito yung mag-explain ng mga diet na low carbs na diet. Kasi makukulangan ng glucose yung body, that's our main energy source. So hahanap ngayon ng bagong pagkukuhanan ng energy yung body natin. So the number one of that is the fats. And then pag na-deplete nahurot na yung fats sa lawas, ano na siya next? Amino acids, pyruvate, and lactate. So ang next na mag-breakdown after the fats will be the proteins. So muna na siya itong makita nga kanagaling pani, tapos bukog na sa kaya nag-breakdown na yung mga proteins din sa body. So saan nangyayari yung gluconeogenesis? It can occur in the liver, in the kidney, and the interstitial epithelium, but except the muscles. Okay? So, andito na naman ito. I believe pina-memorize ito during your biochem days, but because you will be taking the board exam, babalikan niyo ito. Okay? So hindi ko na siya isa-isahin ang pathway. I hope you can still remember this, like glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and then it will be divided into two by the aldolase. We have the dihydroxyacetone phosphate, and then, glyceraldehyde. Dalawa ito, dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, until take note that the end product for our glycolysis is the pyruvate. And then pyruvate will be converted into acetyl coenzyme A, wherein it will be used for the Krebs cycle to produce more ATPs. Okay, so ayan, ganun ka raming steps ang pagdadaanan ni glucose for us to have the energy. Pero kung gluconeogenesis, baliktad, from the pyruvate, pabalik doon sa glucose. Okay, pabaliktad. Aside from the names of the chemical substances there, you also have to be familiar with the different enzymes. Tabang mga langit. Kaya ninyo na laban Japan, ipapilit na sa mga dingding, isulat sa Manila paper. Manila paper lang kay para barato. Kaya mahal mo ng kartulina. Ay, butang sa Manila paper, ipilit sa dingding para para ma-memorize ang glycolysis. And at the same time, since baliktad lang sila ng gluconeogenesis, isama nyo na si gluconeogenesis class. Ang kaibahan nila class, most likely the enzymes. Actually, hindi din lahat ng enzymes yung mga enzymes dyan na iba magkapareho lang. Basta, ang problema ko, naghimo mo sa Manila paper, niya pagkahuman, tama na Jessa, gisulat, ipadikit sa Manila paper, niya giagi-agian lang dito, wala gi memorize. Say sa pagbuhat Manila paper kung wala gi hunungi sa ninyo no, tapos kadugayan og tan-aw ma-memorize siya. Okay? So please include that sa inyong ginastudihan. Okay? Basic glycolysis, it's breaking down the glucose to produce energy. Okay? Energy dyan. Other name for glycolysis is the Embden-Meyerhof pathway. It's a board exam question. Take note. In one glucose molecule, isang glucose lang yon, makakakuha tayo ng 36 to 38 ATPs. If it undergoes complete oxidation. Pag ang isang glucose lang ito, how much more kung daghan kaya itong gikapon. So daghan kaya energy ani. Kaya 36 to 38 ATPs ang ma-produce. Okay? So ito yung pathway ng glucose na hindi kasama yung gluconeogenesis. Okay? Glucose hanggang pyruvate pa rin But you can actually produce pyruvate through the pentose phosphate pathway. Pwedeng yung take note class, yung glucose, pwede siya actually hindi mauwi into pyruvate. Pwede siyang lumiko from the glucose 6-phosphate through the enzyme glucose 6-phosphate dehydrogenase, papasok siya sa pentose phosphate pathway. And sa pentose phosphate pathway, maraming makuproduce dyan, including the ribose, the sugar that we need for the formation of our genetic material. But aside from that, pwede siyang bumalik actually sa glycolysis pathway if you have the extra cellulose 5-phosphate. Pero dito na siya dediretso sa glyceraldehyde 3-phosphate. Okay? So take note na on the first part of the glycolysis class, ATP will be used. Hindi muna magpo-produce ng ATP, gagamit muna ng ATP. Okay? So magpo-produce class ng ATP on the latter portion or the latter part of the glycolysis. So merong dalawa dyan. And then saan galing yung 36 to 38? Class, take note, complete oxidation. So yung 36 to 38, merong iba, this is coming from the TCA or the Krebs cycle. Okay? Basta 36 to 38 yan. Krebs cycle, and then you still have the electron transport chain. And that is for the anong tawag dyan? Nakalimutan ko tuloy. Yung may oxygen na glycolysis, complete oxygen source, pyruvate, and then papasok siya sa Krebs cycle. Pero kapag kulang ang oxygen, instead of going into the Krebs cycle, pyruvate will be converted into lactate. And lactate or the lactic acid will be accumulated in the body, and yan yung explanation sa mga pamaol. Okay, sa mga pamaol class. Kapag nag-exercise ka, again, pag nag-exercise ka and then kulang ng hangin, kulang ng oxygen, kulang ka ng oxygen, hindi ka nasanay mag-exercise ng heavy exercise talaga. Yan, ikalit lang kag-exercise. Syempre, imong body kulang oxygen. Pag mag-breakdown ang glucose natin sa body, marami dun yung magfo-form ng lactate. And then yun yung masustore sa mga kung asa nga part kung asa siya nga part ma-store, and then yun yung mag-cause ng pain. Mga pamaol sa kamot, pamaol sa tiil, kulang na siyang oxygen. Okay? And pag naging lactate kasi yung glucose class, it's a very weak source of energy. So aside from the pain, kapoy imuhang lawas, kapoy imuhang muscles. Dili man good siya good source of ATP. So hindi siya good source of energy. Okay? Now this is class for the glycolysis alone ha. For the aerobic, ito pala yung hinahanap ko na term. Pag may oxygen, that's aerobic. Pag aerobic yung oxidation or yung glycolysis lang, hindi kasama yung TCA, hindi kasama yung Krebs cycle, 1 glucose unit will produce 6 to 8 ATPs. Pag anaerobic, ito yung sinasabi ko kanina na hindi siya good source of energy kasi it will just produce 2 ATPs. Dito lang siya sa lactate. Okay? And then, take note, pag may nakita kayo class sa pathway na FADH2, this is one of the source of ATP. 1 molecule of FADH2 is equivalent to 2 ATPs. So again, source pa rin siya ng ATP natin. Okay? And then sa Krebs cycle, you have kasali yan siya sa ilagay niyo sa Manila paper. And pilit niya sa inyo mga ding-ding. You will notice sa Krebs cycle class, may mga arrows dyan pointing into the production of either NADH, FADH, and GTP. Yan yung actually source natin ng oxygen. You have to be familiar also with the number of ATPs na ma-produce dyan. Kapag ka NADH, 3, FADH, 2, and GTP, 1. Okay, other name for our Krebs cycle is tricarboxylic acid cycle or the citric acid cycle. And another board exam question, it can produce 12 ATPs per cycle. Per cycle, ito ha, isang cycle, 12 ATPs, lahat yung ma-form. So, depende na kung ilang cycle yung pagdadaanan niya. Yan yung total ATPs na na-form lahat-lahat. So, ito na yung ating Krebs cycle class. Medyo complicated lang siya tingnan kasi sinama yung mga structures ng mga chemicals dyan. Pero yung mga naka-violet, ayun, from the acetyl coenzyme A, citrate, isocitrate, alpha-ketoglutarate, and then tingnan saan yung mga na-form na NADH, FADH, or the GTP, paki-count yung number of ATPs. Ito yung corresponding number of ATPs per substance. Okay? Time na. I'll just end siguro with the G6PD deficiency. If I still have time, I'll just be recording the discussion for the lipids. Okay, so after that Krebs cycle, we have the electron transport chain. I-summarize lang natin yung mga important lang sa board exam. We have the final electron acceptor. Electron transport chain kasi ito, magta-transfer step by step ng electrons. And the final electron acceptor is the oxygen. Pag natanggap na yon, the final product will be water. Okay, that's the final product of our electron transport chain. So ano yung mga dapat niyong tandaan sa electron transport chain? Isalin nyo din siya sa Manila paper. Yung mga complexes dyan, though hindi ko nasali sa dito pala yung complex 2, paki-research ng complex 2, may pangalan din yung complex 2. Kasi step by step, from complex 1, the NADH plus the coenzyme Q reductase, tapos mag-jump yan siya sa complex 2, bakit hindi siya nasali dito? Tapos complex 3, paki-take note ng name ha. Naka-encounter ako ng question asking neto, mga names ng complexes sa electron transport chain. Ang type of question nun is, which of the following pair is not correct? Complex 1, tapos yung name, complex 2, and then yung name. Okay? So, pasali yan siya sa pag-aralan nyo and i-familiar nyo. And then lastly, we have the pentose phosphate pathway or the PPP. What are the goals? The goals of the pentose phosphate pathway is first to produce ribose. Sabi ko ito kanina, nagpo-produce siya ng sugar for the product for our genetic material. And then to produce an ADPH also, so that includes production of energy also, and interconvert pentoses and hexoses. So from glucose, maging glucose 6-phosphate, and glucose 6-phosphate, which is a hexose, it's a 6-carbon sugar, will be converted into ribose, a 5-carbon sugar or a pentose. So yan yung goals ng pentose phosphate pathway, or also called as the hexose monophosphate pathway. Now we have shuttles for our pentose phosphate pathway. Ang tandaan nyo lang for the shuttles kung ilang NADH yung kaya. So we have glycerol phosphate 2 and the malate aspartate 3 NADH yan. Saan may pag-memorize ng pentose phosphate pathway? Naglisod na sa glycolysis tapos nag-apil-apil pa ni si pentose phosphate pathway. Hindi naman ito na-ask talaga sa boards. Ang ina-ask sa board exam, na-summarize ko na ang goal, ang other name ng pentose phosphate pathway, and yung mga shuttles involved sa pentose phosphate pathway. Okay? Mga shuttles yan. Naman, gamit ng shuttle, syempre mag-carry. Ito yung kini-carry nila, 2 NADH, 3 NADH. Okay? Now, si pentose phosphate pathway, magsa-start yan siya kay glucose 6-phosphate. Ito. Okay? And it needs the glucose 6-phosphate dehydrogenase enzyme para magpatuloy yung pentose phosphate pathway, mabuo yung mga ribose, makuha natin yung mga kailangan natin na chemicals here, yung NADPH for the energy. Kailangan natin si glucose 6-phosphate dehydrogenase. Our problem, may mga tao may G6PD deficiency. So it's actually a problem on the pentose phosphate pathway, and ang consequence of their deficiency of the enzyme is hemolytic anemia. So remember, yung ating goal talaga is to produce the ribose for the genetic material, but yung problema, hindi mabubuo ng exactly yung RBCs nila. Kaya naghe-hemolyze, they have hemolytic anemia, especially in the presence of oxidizing agents like the sulfa drugs and our anti-malarials. So be careful with giving food or drugs for the G6PD deficient na mga patients. Again, yung mga sulfa drugs and anti-malarials, pag binigay niyo yan sa kanila, masamutan ang pag-hemolyze sa ilahang RBCs. Anong kulang? The enzyme involved here in the pentose phosphate pathway that will degrade glucose 6-phosphate to all the other chemicals here in the pathway that are necessary for, number 1, our genetic materials and all the other na mga chemical processes sa body that involve this one. Okay? So I think dyan ako mag-i-end. And again, kapag may time ako, I will be making a recorded discussion for the lipids. Actually, konti na lang yun. Okay? Ang final exam coverage will be the whole biochem thing. Whole biochem yan. And then, busy sila sa GC for the compre. My gosh. Napay compre. Wala na pay time. Napay compre.